Okay, everybody. I know now you've gone through watching kind of a ridiculous video in the check-in. You went through a somewhat of a basic simulation. Hopefully, you were able to figure that out. Hopefully, my video I made beforehand helped you negotiate that. And then you watched a really, I thought, a really well-made animation of the whole process of protein synthesis. And we learned that central dogma or central truth of making proteins is that the DNA, deoxyribonucleic acid, the blueprint for all the proteins in our body, codes for this thing called messenger RNA. And then messenger RNA actually is what is used to translate that code into protein. And then protein goes back and does everything. I mean, RNA polymerase is a protein. We're going to find out that the receptor for COVID-19 is a protein. And these things relate to organic chemistry and they relate to functional groups. But what I wanted to do is just break down that first process really simply, the process of the DNA to the messenger RNA. And you notice last question I asked you, was there anything that you learned in this video that you did not see in the simulation? And you might have seen me starting to reveal to you what that DNA code looks like. Okay, so I want you to write this down in your notes. This might be oversimplified, but what I want to talk about is in this video some of the organic chemistry that underlines what we just saw. So let's say we have a little person right here. Go away there. So you get a little person right here. Okay, a little person. And this person we know is made out of cells, right? Eukaryotic cells, okay? So if we looked at one of these cells, inside one of those cells is this nucleus. It's got a nuclear envelope around it, and that's protecting the blueprint. It's protecting the information that's going to make the proteins which make us, and we need to protect that. So if we look inside there, what is all that stuff? Okay, well, all that stuff we know is DNA, and it's a double helix formed into these 23 chromosomes. And you might have saw something in the last video. You saw that double helix is actually connected by these chemicals. We call these chemicals nitrogenous bases. And why they're bases, why they tend to have pHs higher than 7 at standard homeostatic conditions in the human body is one thing, but they are made of nitrogen and they are very important. And it turns out that they have a very systematic way of connecting. And it turns out these two strands really aren't even connected. There's an intermolecular force or an attraction between them. And it turns out that you have what's called base pairing. So every single time on one strand you have an adenine molecule, and we use an A for that, that is always going to pair up with a thymine molecule. Now, not thiamine, which is vitamin B, thymine. Okay, so anytime you have a T on one, you're always going to have an A on the other. If you had an A here, you'd have a T here. Okay, and then there's another kind of base, two kinds, and that's guanine and cytosine. And they always pair with one another. So every time you have a G, you have a C. And every time you had a C here, you can have a G. And let's say we had a G here, we have a C. So we can start decoding the strand. We know that on one side, if you have a T, we know that the other side is going to be an A. If we have a C, we know the other side is going to be a G. If we have an A, it's going to be T. If it's a C, it's going to be G, T, A, G, C. Okay, so these have chemical structures, and that's really the focus of this. But these bases make up DNA, and we're going to find out later that for every 3 bases, you end up getting 1 amino acid. And this thing here is called a codon. So the organization of 3 bases is going to end up coding for amino acid. So for example, AGT, that sequence, or the sequence TCA, because remember, TCA is the exact same thing as AGT. Okay, it's its complementary base. They're going to give us a specific amino acid. So what we can do is we can start looking at sequences of genes. So let's say we have AGT, and here we have GAC. That might be amino acid 2. And then when we know when amino acid 1 and amino acid 2 come together to make a protein, that protein does something. So that's the essence of the code. Okay. Well, you saw in the video, though, that when we have a strand of DNA, okay, let's take this same strand that we had before, that strand of DNA is transcribed. There's a process of transcription that happens where the DNA is turned into RNA. In this case, we call it messenger RNA because it's sending the message as to what protein we want. So I want to take this same strand here. So we had A, G, T, G, A, and then we had C here. So let's extend this one more time. And I believe that was a C. So we know immediately that this must be T, this must be C, this must be G, this must be C, this must be T, and this must be G. TCG, CTG, TC. Oh, I'm sorry. I messed up here. I made a boo-boo. This must be A. Sorry about that. Okay, so AGTGAC, AGTGAC, AGTGAC is the same thing as TCA, CTG, TCA, CTG. So what we know, though, is that this is just the blueprint. That needs to be transcribed. And we had that big, RNA polymerase molecule come on here with those transcription factors, and it did this crazy thing. And what it ended up doing, though, is that it ended up transcribing a single strand. So we had a single strand that ended up being transcribed from one of these strands of DNA. Okay, so what we ended up with is a messenger RNA strand. Let's say we transcribe the top. So the A, okay, in this case here, would be transcribed in messenger RNA to a U. So when we have DNA to messenger RNA, T is always converted to U. And that's one of the ways we know we have messenger RNA. There is no more thymine. Thymine becomes uracil, but everything else stays the same. So we know here that that G must be a C, and that T here, I'm sorry, I am having brain fartage. That this T here is going to become an A. We still have A. And we know that this G here is going to be a C. And we know that this A here is going to be a, what's that A going to be? Is there T? Nope, it's going to be a U. And then we have this C here, which is going to be a G. So notice that the messenger RNA strand that came from that single strand of DNA looks just like the complementary strand of DNA, except for every time there's a T, there is a U. So here we have UCA, CUG. And that's that messenger RNA strand. And this is really the process of transcription. This thing is now going to leave the nucleus, and each of these 3 codes is going to code for different amino acids, and this is going to make a protein which is going to do something, and we'll be talking about that later. But I want to draw your appreciation now for what are these things, okay? So we know that in DNA, A always goes with T, and G goes with C, and T goes with A, and G goes with C, and A goes with T, and C goes with G. We know this, and this goes on forever and ever and ever, and allows us to predict the code, okay? And we know in that code, when one strand of that is transcribed, meaning we take one strand of that and we create a single strand messenger RNA, that this process is called transcription. And we know that during that process, for some reason, the T's are switched with U's. And I think that's probably done so that the cell knows we're dealing with some messenger RNA. But what are these? Okay, what are these? Let's take a closer look at them. So they fall into two categories. We have the purines, and we have the pyrimidines. Okay, and the purines and the pyrimidines have some functional differences. First, if you notice, the purines have this pentene ring and this hexane ring, and the pyrimidines do not have that. And the purines are A and G. Now, we know that A binds with T or U in RNA, but the purines themselves have this structure. So we have A and we have G here. And here we have the pyrimidines, thymine, T, cytosine, C, and uracil, U. Okay, and in terms of pairing, we know that A is always going to pair with T, but A is going to pair with U in messenger RNA, and G is going to pair with C always. But these are really the backbones of DNA. You can think of the DNA strand, okay, as being like this and like this here, and A is pairing with T here, and G is pairing with C or U. So what I'd like you to do is I'd like you to pause this video, and I'd like you to identify really all the functional groups that you see here in adenine, all the functional groups you see here in guanine, all the functional groups that you see here in thymine, all the functional groups you see here in cytosine, and all the functional groups you see here in uracil. And really, I'll be honest with you, let me see here. I only see 1, 2, I only see 2 functional groups. Call me crazy. So really, I wanted to finish this whole lesson where we started the year with organic chemistry, and these bases really are provide the functionality. They provide the code, the purines and the pyrimidines of DNA, ultimately RNA, and then ultimately protein.